Okay, so before we can try to reconstruct this science paper, we first need to do something called informatics work. We have the yeast gene evolutionary rate data in this file sce.spa.kx.csv, and that's the evolutionary rate data. We also had the protein interaction network data in pairs. That's this file called pairs.csv. And we first need to remember figure 1 is basically number of interaction over evolutionary rate. We first need to merge these two data sets. I'm merging this kind of large data set. I guess that's what we call informatics work. And since this is biological data, we call that bioinformatics. I'm not the one to make up the word. I'm just guessing here. Okay. So we have two data sets. We want to merge them. So let's first read into these two data sets. Okay, very good. And data is, here's data. And basically, we have the gene name, evolutionary rate. That's the data. What's the pairs? Pairs is a pairs of systematic gene names. So we need to merge these two data sets. We need to generate a table where we have number of gene interactions and then the evolutionary rate, and then we can redo figure 1. And that's the growth fitness data, and let's ignore that for the time being. And so first, how do we find out the number of interactions from the pairs network data? And the way I try to do it, I basically, for every gene in that pairs list, the occurrence, if that gene occurs twice, that means that gene is in 2 pairs. The assumption is that list has no self-interaction pairs. So if the gene interact with itself, we remove it. We don't consider that as pairwise interaction. So I have removed all those self-interacting pairs, and then the remaining data, the number of occurrence of every gene, every social security number, that means how many interactions that social security number has. Right? So if how many friends do you have on Facebook, and if you put your friend list on Facebook in your pairwise data, how many times your social security number occur in that list is basically how many friends you have. Does that make sense? Doesn't seem okay. I'm going to come back later, but right now let me just move forward to finish this. So once I basically generated the list, and I put that list into a long vector, then I use this command called table. A table command basically counts the occurrence for every character in that array. And this table command basically calculates the number of interactions for every gene. And actually, basically that's it. These two commands will calculate the interaction for every gene. And the trick is now we need to put the number of interactions with the evolutionary rate in the same data frame. Once we put in the same data frame, we can do a scatter plot and do a linear regression. That's the figure 1 of the Fraser science paper. And here, in order to do that, I put into a data frame. Here, to do a data frame network, I make sure the data frame is in a character here. So right now it's a factor. I think sometimes the factor can be confusing, so I change it to a character. So everything is a character. And I remove the first column -1 means I remove the first column. And here I check, here is a consistent check to see, I now have two data sets, which I call net. That's the number of interaction per gene. And then I have the evolutionary rate data. I want to merge these two data. So for every gene here, I want to find it out here. So for example, YAL007C, YAL007C here, the evolution rate is 0.01. I want to put this number, I want to merge these two data sets. So this intersect actually doesn't do the merge, but what it does is check how many genes are shared between these two data. So intersect say I want to see how many genes are shared between net ID and the data frame.orf. If we go back, data, the ORF, in the data, the ORF name basically the social security number for each gene. And if we go back to the net, the ID here is the social security number for the each gene. So basically, I'm trying to say in this list of social security number here, how many of them are actually the same. Those I can only move those data which are the same. If they are not covered, basically they cannot be merged. Right? So only some of the gene doesn't have evolutionary rate, some of the gene doesn't have interaction data. Remember, those are both experimental data. Sometimes we do not cover them during experiment. Or genes in gene sequencing, sometimes we're also missing them. And the evolutionary rate right now is more complete, but I haven't updated it yet. So the evolutionary rate data is also incomplete for this analysis. Okay. So basically, if you try to, without merging the data, if you want to do a linear regression if you do a linear regression, see does the evolutionary rate correlates with gene frequency, this won't work because they are in two different data frames. You have two different spreadsheets. So this command will not work because the data has not been merged yet. We need to merge the two data sets. And this can be done by this command called the match command. So the match command, what the match command does here is it matches the gene name in data, which is the evolution data, to the net gene ID. After so every number here will match with net gene ID. Remember, and then I put this inside of the square bracket. The square bracket is the position of the column. So that's the position of column in net.frequency. And then put this whole thing into data.degree. Data is the evolutionary data. So basically, net is the interaction data. I match it with the social security number in the evolution data, and then that's the position of those match in the network net data, and then put this frequency data into the evolution data, and I call that a degree. That's basically the connection degree. This actually finished the merging of the data.